You know, when we look at a city's skyline at night, we just see millions of lights on. It looks so stable, permanent and peaceful.
Right. Very calm on the surface.
Exactly. But beneath the streets, there is this chaotic, nonstop symphony going on. You've got power plants burning fuel, transformers stepping down voltage, and thousands of invisible switches routing electricity to exactly where it's needed.
Down to the exact millisecond.
Yes. And if you think about it, well, your own body is running the exact same way right now.
It really is. It's just a phenomenal balancing act. And today, we're going to turn that lens inward to look at the microscopic power plants running you.
Yeah, the cellular engines.
Right. So for you listening, whether you're, say, a medical student prepping for boards, or maybe a young professional brushing up on foundational sciences, or just an insanely curious learner, our mission today is to completely demystify human metabolism and cellular biochemistry.
And we are drawing heavily from a really great source for this deep dive, which is Oraby's Illustrated Reviews of Biochemistry.
Yeah, it's an excellent text. We're going to use it to map out how your molecular engines actually function, how they adapt under stress, and what happens when the machinery actually breaks down.
Right. So to start off, we hear the word energy thrown around constantly in daily life, right? Like energy drinks, energy levels.
Oh, all the time.
But biochemically, what are we actually talking about when we say a cell is using energy?
Well, we're talking about bioenergetics, and specifically the concept of free energy, which is often denoted as delta G.
Delta G. Yeah.
So free energy is basically the useful energy available to do actual work in the cell. Every chemical reaction in your body essentially falls into one of two camps. First, you have exergonic reactions.
Okay, exergonic. Meaning?
Meaning they have a negative delta G, so they release energy. Think of like a boulder rolling down a hill. It happens spontaneously and gives off kinetic energy.
Makes sense.
On the flip side, you have endergonic reactions. These have a positive delta G, so they require a substantial input of energy to happen.
Like pushing that heavy boulder back up the hill.
Exactly. Things like building a muscle fiber or firing a nerve impulse. Those are endergonic. They cost energy.
But you can't just push a boulder up a hill with good intentions. I mean, you need a mechanism to capture the energy from the falling boulder and, well, transfer it to the one you're pushing up.
Yes, and that middleman is the ATP-ADP cycle. ATP stands for adenosine triphosphate. It's the universal energy carrier across, well, all human cells.
The universal currency.
Right. Its entire job is to link the energy-producing exergonic reactions to the energy-consuming endergonic ones.
Okay, let me put it this way, because a financial analogy really helps ground this for me. If a molecule of glucose from, say, a sandwich you just ate is a high-value gold bar, the cell can't just walk into a corner store and spend it everywhere.
No, it's too bulky. It's the wrong currency entirely.
Right. The cell has to take that gold bar to the metabolic bank, break it down, and cash it in for ATP. Because those ATP molecules are the $1 bills the cell actually uses to do the day-to-day biological work.
That's a great way to look at it. And the mechanism behind why those dollar bills hold value is really elegant. ATP contains 3 phosphate groups lined up next to each other.
And phosphates are negatively charged.
Highly negatively charged. So you're cramming 3 negative charges closely together, and they strongly repel each other. It's like compressing a heavy spring.
Oh, wow. So there's all this potential energy just waiting to snap.
Exactly. When the cell snaps off that third phosphate converting ATP to ADP, adenosine diphosphate, the spring releases. And that burst of chemical energy is what powers the cell's machinery.
That's amazing. And there are other molecules that hold even more tension in their springs, right? Like creatine phosphate in our muscles.
Yes, creatine phosphate acts as a rapid response energy reserve. When it transfers its phosphate to make new ATP, it gives off about negative 10.3 kilocalories per mole.
Wow, negative 10.3. That sounds like a lot.
It is a massive cellular energy release. It's designed to keep your muscles firing during those first few seconds of an intense sprint, just before the rest of metabolism can catch up.
So to see how the bank actually processes the deposit, let's follow the gold bar. Let's trace the journey of a glucose molecule. Say you eat some complex dietary starch. The digestion actually starts the second food hits your mouth, right?
It does, yeah. An enzyme in your saliva called salivary alpha amylase begins cleaving the large carbohydrate chains immediately.
Which is right there in the mouth.
Right there. Then the process pauses in the acidic stomach, but resumes in the small intestine, where pancreatic alpha amylase finishes the job.
Breaking the starch down into pure single glucose molecules.
Exactly, so they can be absorbed into the bloodstream.
Okay, so once that glucose enters a cell, it goes right into the fluid of the cell, the cytosol. And that is where glycolysis begins, right? The first step of breaking down the glucose.
That's right. Glycolysis happens right in the cytosol.
But oxygen isn't always present in every tissue, or at least, not in high enough quantities during heavy exercise. So does the cell just stop processing glucose if it's holding its breath?
Not immediately, no. Glycolysis itself, the act of splitting a 6-carbon glucose into 2 3-carbon molecules called pyruvate, it doesn't strictly require oxygen.
Oh, interesting.
Yeah, if a cell lacks oxygen, it performs anaerobic glycolysis. It takes that pyruvate and converts it into lactate, or lactic acid.
But why bother making lactate? I thought the goal was to make ATP.
Well, the primary goal of converting pyruvate to lactate isn't actually to make energy. It's a rescue operation.
A rescue operation. How so?
So glycolysis requires a steady supply of a coenzyme called NAD+ to keep running. When oxygen is low, NAD+ gets depleted really quickly. By converting pyruvate into lactate, the cell chemically regenerates NAD+ active.
Ah, okay, so it's basically clearing the factory floor so glycolysis can just keep churning.
Precisely. But the trade-off is that the energy payout is incredibly small. You only get a net yield of 2 ATP per glucose molecule.
Which perfectly explains a really fascinating fact about red blood cells. Because mature red blood cells are the vehicles that transport oxygen to every corner of the body. But they completely lack mitochondria, which are the organelles that actually use oxygen to make energy.
It's a brilliant evolutionary adaptation, honestly. Think about it. If red blood cells had mitochondria, they would act like a delivery driver eating the pizza on the way to the house.
That's hilarious. They just eat all the cargo.
Exactly. They would consume the oxygen payload before they ever delivered it to your tissues. Because they lack mitochondria, they rely entirely on anaerobic glycolysis.
So they just churn out those 2 tiny ATPs and convert pyruvate to lactate just to survive the trip.
Yeah, it's incredibly efficient for their specific job.
But for a typical cell, like a muscle or a liver cell that does have mitochondria and plenty of oxygen, that pyruvate takes a completely different path, right? It goes inside the mitochondria and enters the Krebs cycle.
Right, also known as the TCA cycle. Inside the mitochondria, the cell completely oxidizes the molecule. It strips away high-energy electrons and sends them through a sequence of proteins embedded in the mitochondrial membrane.
And that's the electron transport chain. Because this is where the energy yield gets exponential, doesn't it?
It really does. As those electrons bounce down the chain, they power pumps that push protons across the membrane. This creates a massive electrochemical gradient.
Like water building up behind a dam?
Yes, and when those protons flow back across the membrane like water rushing through a hydroelectric dam, they physically spin an enzyme called ATP synthase.
So what's the ultimate payout from this aerobic pathway?
A staggering 38 ATP per single glucose molecule.
Wow, 38 versus 2.
Yeah, compared to the 2 ATP from the anaerobic route, the mitochondria are just the absolute workhorses of human metabolism.
Okay, so we know how the body caches in glucose when times are good. But we don't just shut down the moment our stomach is empty. I mean, if I skip lunch, my heart doesn't stop beating. So how does the body keep the lights on when the primary fuel runs out?
Well, this is where we look at the body's backup generators.
Yeah.
Human metabolism operates on what we call the fed starvation cycle.
The fed starvation cycle.
Right. You are constantly shifting gears based on nutrient availability. When you enter a fasting state, say 6 or 7 hours after a meal, your blood glucose begins to drop, and the liver steps up to the plate first.
Okay. What does the liver do?
It stores a complex, highly branched carbohydrate called glycogen. The average liver holds about 120 grams of it.
But wait, 120 grams sounds like a tiny buffer for an entire human body.
It is tiny. The liver rapidly breaks this glycogen down into free glucose and releases it into the blood to maintain baseline levels, mostly to keep the brain fed.
Right, because the brain needs a lot of fuel.
A huge amount.
Yeah.
Because the brain is such a greedy consumer of energy, that liver glycogen completely runs out after just 12 to 18 hours of fasting.
And once the liver's direct reserves are gone, the body has to start manufacturing glucose from scratch, right? Gluconeogenesis.
Yes, gluconeogenesis literally translates to the creation of new glucose. The liver starts gathering non-carbohydrate scraps from around the body.
What kind of scrap?
Well, it takes lactate returning from the muscles through a pathway called the Cori cycle, or it starts stripping amino acids from muscle proteins and reverse engineering them back into glucose.
But breaking down your own muscle tissue to make glucose, I mean, that isn't a sustainable long-term survival strategy. Eventually, you just waste away.
Absolutely. So the body has to pivot to its largest reserve, which is lipid metabolism, fat.
Right, our fat stores.
Adipose tissue stores fat in the form of triacylglycerols. When insulin drops during fasting, enzymes break these down into free fatty acids, which flood the bloodstream. And the energy density of these fats is incredible.
Let's quantify that because the contrast is wild.
Yep.
We established earlier that one molecule of glucose gives you 38 ATP. What happens when the mitochondria process a fat molecule?
Okay, let's take palmitic acid, which is a standard 16-carbon fatty acid. It enters the mitochondria and undergoes a process called beta oxidation.
Beta oxidation.
Right. Because a fatty acid is essentially a long chain of carbons totally saturated with hydrogen atoms, there are far more electrons available to be stripped away and sent to that electron transport chain we talked about.
So what's the yield?
The yield from one molecule of palmitic acid isn't 38 ATP, it's 129 ATP.
129. It's basically an ultra-high capacity battery.
Precisely. The liver eagerly oxidizes these fatty acids. But as the liver burns fat at this massive accelerated rate to power gluconeogenesis, it produces an overflow byproduct.
Ketone bodies, right?
Yes, ketone bodies. This process is called ketogenesis. The liver can't actually use these ketone bodies itself, so it ships them out into the blood.
Now, I need to push back on this a bit, because a core rule of neurobiology is that the brain relies exclusively on glucose for fuel. The blood-brain barrier blocks fatty acids from entering.
And is generally true, yes.
So if the liver runs out of glycogen in 18 hours, and gluconeogenesis can only produce a limited supply, how does the brain survive a week or a month of starvation?
That's the amazing part. The brain does heavily prefer glucose under normal conditions. However, if starvation persists for a few weeks, the brain undergoes a profound metabolic adaptation.
Oh, really? It adapts?
Yes. It begins expressing enzymes that allow it to use those ketone bodies as its primary fuel source. It's a critical survival mechanism.
Wow.
By switching the brain over to running on ketones, the body dramatically reduces its overall need for glucose.
Which means it doesn't need to break down as much muscle protein to feed the gluconeogenesis pathway. It spares the structural integrity of the body.
Exactly. It slows down muscle wasting and extends survival time during famine significantly.
The body acts like a highly advanced hybrid engine. It seamlessly transitions from an electric motor to a gas generator depending on the terrain, all to protect its most vital hardware.
That's a perfect analogy.
But, who is driving the car? The liver doesn't just spontaneously decide to stop storing glycogen and start burning fat.
No, the drivers are the hormones. These chemical messengers orchestrate the entire metabolic symphony. And to understand how they work biochemically, we categorize them by their mechanism of action.
Right, how they actually deliver their instructions to a cell, because they don't all knock on the door the same way. Let's start with group I hormones.
Sure, group I hormones include your steroid hormones like cortisol and the thyroid hormones. The key defining feature of group I is that they are lipid soluble.
So they don't need a doorway.
Exactly. They dissolve right through the fatty outer membrane of the cell, travel straight into the nucleus, and bind to intracellular receptors.
And what do they do once they're in the nucleus?
They physically bind to the DNA. They alter transcription, turning the synthesis of new enzymes and proteins on or off. Because they're building new machinery from scratch, their effects take hours or days to fully manifest.
Okay, but then you have group 2 hormones, which are protein or peptide hormones. They're water-soluble. So if they try to cross the fatty cell membrane, they bounce right off. They're completely locked out. My mental model for this is a delivery driver who arrives at a high-security office building.
Okay, I like where this is going.
They aren't allowed inside. They just walk up and press the buzzer on the outside panel. That buzzer is the cell surface receptor.
Right.
When the hormone presses it, a security guard who is already inside the building hears the chime, grabs the message, and runs around altering the cell's machinery.
Spot on. And the biochemical term for that internal security guard is a second messenger. Right. The most prominent second messenger we study is cyclic AMP, or CAM-AMP.
Okay.
The hormone never enters the cell. It binds the receptor on the surface, an enzyme on the inside of the membrane activates, cyclic AMP levels spike, and within seconds to minutes, existing enzymes in the cell are phosphorylated and turned on or off.
So it's a super rapid response system.
Very rapid.
And the two major rival delivery drivers utilizing the system are insulin and glucagon.
Yes. Insulin is the ultimate building hormone. When you eat a meal and blood sugar spikes, the pancreas secretes insulin. It binds to cell receptors and acts like a key, unlocking transport channels to pull glucose out of the blood and into the muscle and fat cells.
And it also stimulates the storage of fat, right?
It does. It stimulates glycogen synthesis and fat storage. Glucagon, on the other hand, is the mobilization hormone.
The rival driver.
Exactly. It's secreted when you're fasting. It rings the cyclic AMP buzzer on liver cells and says, break down the glycogen, release the fat. We need energy in the blood now.
There is a really fascinating detail regarding insulin dynamics, though. If someone's diet causes their insulin levels to be chronically constantly high, say in cases of severe obesity or chronic overeating, the cells eventually get overwhelmed, right?
They do. They actually start physically removing the insulin receptors from their surfaces.
That's crazy.
It's a process called downregulation. The cell's basically putting in earplugs because the insulin signal is deafening.
Which is the exact biochemical root of insulin resistance. The pancreas is shouting, but the cells refuse to listen.
Right, and that transitions us into the most critical part of this discussion. What happens when these incredibly precise systems break down?
Yes, let's look at system failures. When the rules of normal biochemistry are broken, the pathology is severe. Let's start with diabetes mellitus.
Okay.
This represents an absolute lack of insulin, as in type 1, or a relative lack in resistance to it, as in type 2. Biochemically, the cascading disaster makes total sense based on what we've covered today.
It really does. Without insulin signal, glucose cannot enter the muscle or adipose cells. It gets trapped in the bloodstream. So blood sugar spikes dangerously high, a condition called hyperglycemia.
Right.
Eventually, the concentration of glucose in the blood gets so high that the kidneys, which normally filter and reabsorb all glucose, simply cannot keep up.
That threshold is about 180 milligrams per deciliter, right?
Exactly, 180.
Once blood glucose hits that number, the kidney's transport proteins are completely saturated. So the excess glucose spills over into the urine, which is clinically known as glycosuria.
Yes. And because glucose is a highly osmotically active molecule, it drags massive amounts of water out of the body along with it.
That osmotic pull explains the profound dehydration and frequent urination polyuria that we see in uncontrolled diabetes.
Right. But the deeper tragedy of diabetes at the cellular level is that the body's tissues are literally starving amidst a sea of plenty.
It's like starving at a buffet.
Exactly. There's energy floating everywhere in the blood, but without the insulin key, the cells are locked out.
So the body panics. It assumes it's in a state of extreme, life-threatening starvation, even if the person just ate a massive meal.
Yes. So it flips the switch to the backup generators. The liver aggressively breaks down adipose tissue. It burns fats at such a rapid, uncontrolled rate that it produces massive, unmanageable amounts of ketone bodies.
And ketones are acidic, aren't they?
They are very acidic. The accumulation of these acidic ketone bodies overwhelms the blood's buffering capacity. This leads to dangerous ketosis and ultimately ketoacidosis.
Which is life-threatening.
Very. It causes a severe drop in blood pH and can easily be fatal. It perfectly illustrates how disrupting one single hormonal signal unravels the entire metabolic integration of the body.
So how do we use this clinically? I mean, how do we peek into this biochemical chaos without doing invasive procedures on a patient?
Well, biochemistry provides us with really powerful diagnostic windows. In diabetes management, an isolated blood sugar reading only tells you what is happening in that specific minute.
Right, but you want to know the long-term trend.
Exactly. When glucose levels are chronically high, those glucose molecules bump into proteins in the blood, like hemoglobin inside red blood cells, and slowly, non-enzymatically stick to them.
Okay.
This is a process called glycation.
So by measuring HbA1c glycated hemoglobin, a doctor can see a permanent physical record of what a patient's average blood sugar has been over the lifespan of a red blood cell, which is what, about 2 to 3 months?
Right, about 2 to 3 months. It's a much more reliable metric for assessing metabolic control.
But diabetes is just one facet of metabolic failure. We also see systemic failures in lipid transport, leading to atherosclerosis.
Yes, another major failure point.
Because fats do not dissolve in the watery environment of blood. If you pour oil into a glass of water, it just floats on top.
Right, they don't mix.
So to move fats around, the body has to package them inside protein vehicles called lipoproteins, specifically VLDL and LDL, which deliver cholesterol from the liver to the peripheral tissues.
And cholesterol is essential for building cell membranes and synthesizing those group I steroid hormones we talked about. But problems arise when there's an excess of LDL particles lingering in the bloodstream.
What happens to them?
Well, the delicate inner lining of the blood vessels, the endothelium, can become damaged from high blood pressure or smoking. These LDL particles slip into the vessel wall and get oxidized by free radicals.
And the immune system views oxidized LDL as a toxic invader, doesn't it? So it sends in macrophages, a type of white blood cell whose job is to eat debris.
It does. The mechanism here is a fatal flaw in the macrophage's programming. Most cells downregulate their receptors when they have enough of something, just like we saw with insulin.
Right, they put in the earplugs.
Exactly. Macrophages, however, possess scavenger receptors that bind to oxidized LDL, but these receptors do not downregulate.
Wait, really? So they just keep eating?
Yes, the macrophage will continue to consume oxidized cholesterol indefinitely.
They gorge themselves until they physically transform into these bloated entities known as foam cells.
Exactly. And the foam cells eventually die from the toxic accumulation. They rupture and spill their necrotic, cholesterol-rich contents into the artery wall.
Oh, wow.
Yeah, this triggers a massive inflammatory response, building the fibrous atherosclerotic plaques that narrow the arteries and ultimately rupture to cause heart attacks and strokes.
It's a normal defense mechanism, cleaning up trash that just doesn't know when to turn off.
Precisely.
And speaking of normal mechanisms failing to turn off, there's the ultimate system failure: cancer and the loss of cellular mortality.
Yes. Every normal cell in the human body has a built-in self-destruct sequence. When a cell accumulates too much DNA damage, or simply reaches the end of its functional lifespan, a state called senescence, it initiates apoptosis or programmed cell death.
It's not a messy explosion, though. It's highly organized.
Highly organized. The cell activates specific intracellular enzymes called caspases. You can conceptualize caspases as a precision demolition crew.
That demolition crew, I like that.
Once a signal is given, caspases systematically cleave the cell's DNA, dismantle the structural cytoskeleton, and package the cell into neat little fragments. Then surrounding immune cells can easily clear them away without causing inflammation.
But cancer cells are essentially rogue elements that have figured out how to evade apoptosis. They mutate the pathways that normally activate those caspases, right?
They do. They turn off the self-destruct switch, allowing them to replicate endlessly with damaged DNA, achieving a terrifying sort of biological immortality.
That is terrifying.
However, because a cancer cell's biochemistry is so fundamentally altered, they often synthesize and leak unique proteins into the blood that normal cells don't produce, or produce in very small amounts.
And we utilize these as tumor markers.
Yes, tumor markers.
Things like PSA, prostate-specific antigen, which can elevate significantly when prostate tissue becomes malignant, or HCG, a hormone normally only seen to support pregnancy, which can spike dramatically in certain cancers like chorion carcinoma.
Exactly. It gives oncologists a biochemical fingerprint to track the presence and burden of the disease. And understanding the baseline, the bioenergetics of ATP, the fuel cycles of fasting, the receptor mechanisms of hormones, it's the only way to recognize the fingerprint of pathology when the machinery breaks.
Well, let's take a breath and recap the incredible journey we've just taken through the body's internal skyline. We started down at the microscopic level, watching the cell cache in its glucose gold bars for the $1 bills of ATP.
Right.
We watched the body brilliantly switch its engines during fasting, relying on the backup generators of fat and ketone bodies to keep the brain alive and spare our muscles.
A true hybrid engine.
Yeah. We mapped the communication networks where hormones act like delivery drivers pressing the cyclic AMP buzzer to change the cell's behavior. And finally, we saw the clinical realities of what happens when the buzzers break in diabetes, when macrophages eat themselves to death in atherosclerosis, and when cancer evades the cellular demolition crew.
It is a dense amount of material to synthesize. So to honor the educational focus of today's deep dive, I want to leave you, our medical student or professional listener, with a specific review exercise to test your integration of these metabolic concepts.
Oh, a pop quiz. I love it.
Exactly. Based on our discussion today, if a patient has a rare genetic defect that completely prevents the synthesis of cyclic AMP inside their cells, which specific class of hormones would fail to function? And what would be the immediate, life-threatening metabolic consequence for this patient during a prolonged fasting state?
Oh, that is a phenomenal question. You really have to trace the group 2 pathway and the glucagon response to figure that out. Consider that your homework.
Let's see who gets it.
And I want to leave you with one final thought to mull over, building on the mechanisms of apoptosis we just explored. Consider this. If cancer cells have simply figured out how to suppress the caspase enzymes that cause normal programmed cell death, could the future of oncology lie not in creating ever more toxic poisons to blast the tumor, but simply in developing a targeted key that flips that switch back on, reminding those immortal cells how to die normally.
That conceptual pivot, restoring normal biochemistry rather than destroying aberrant tissue, is precisely the frontier of modern targeted therapy.
Yeah. It's incredible. Well, keep questioning the incredible machinery inside your own body. Until next time, keep those metabolic engines running, and we will catch you on the next deep dive.